ATP synthase, it is a protein assembly in the inner mitochondrial membrane. It is also called as fifth complex. So it is a multi-subunit transmembrane protein that has two functional units named as FO and F1. So as we said, it is a multi-subunit transmembrane protein with two subunits, FO and F1. FO unit, it is hydrophobic in nature, and this FO unit, it spans the inner mitochondrial membrane. This acts as a channel through which protons enter into the mitochondria. It has 4 polypeptide chains and it is connected to F1. F1 complex, it is hydrophilic in nature and this is tightly bound to the FO subunit. It will project into the mitochondrial matrix from the inner mitochondrial membrane. This has catalytic site for the synthesis of ATP. The F1 unit, it is water-soluble peripheral membrane protein. It has 9 polypeptide chain, each subunit having different binding capacity and catalytic activity. The F1 has 3 conformational states for the alpha-beta functional unit. O state, it does not bind to the substrate or product. Then in L state, there will be loose binding of substrate and product. Then in T state, there is tight binding of substrate and product. So O form is the open and it has no affinity for substrate. In L form, the substrate binds with less affinity. Then in T form, it binds substrate tightly and catalyzes the ATP synthesis. So the bond is synthesized in the T state and ATP is released in the O state. Initially, the ADP and inorganic phosphate, it is loosely bound to the catalytic site on F1. Then as the FO accepts proton, the affinity for ADP is increased. So the L form, it is converted to the T form. So ATP synthesis occurs in the T form and T state, it converts back to the O state when ATP is released.